So I'm actually very excited about being here. I'm very new to biochar, so this would be my first biochar talk ever, and I was very much all about wind as soon as I heard about it too. And I'm going to be, my background, just so you know, is more in plant defense and how the environment and climate affects plant secondary metabolism. Now I've kind of incorporated in here into looking at the effects of soil biochar amendment on wheat, resistance to Fusarium head blight, and mycotoxin contamination. So to start off with, Fusarium head blight is one of the most devastating plant diseases worldwide. Production losses have been estimated to be about 50%, which is quite high. And epidemics within the United States result in billions of dollars in economic losses. So it's a serious plant disease. It is caused by the fungal plant pathogen called Fusarium graminearum, depicted here both on culture media, on culture media as well as on the wheat head itself. So when the fungus invades the wheat spikelet, it causes premature bleaching, and as it spreads throughout the head, so does that bleaching, that symptoms. So it basically looks like it's senescing more quickly. You can see the infected head here, visualizing that. And what happens is the grain begins to shrivel and becomes discolored. So Fusarium graminearum not only destroys the grain yield and quality, but it also produces trichothecene mycotoxins, which are quite hazardous to human and animal health. Trichothecene mycotoxins are fungal secondary metabolites. And in particular, trichothecenes function as a virulence factor that disables the plant defense mechanisms and leads to the increased spread throughout the wheat head. But these chemical compounds function by inhibiting plant protein synthesis. So as you can imagine, they are quite deleterious to human health if they are consumed within the grain or grain products. Deoxynivalenol is the most predominant trichothecene produced by Fusarium graminearum, and is also frequently called vomitoxin for the effects it has if you do consume it. So you can understand why it is so closely regulated and considered such an issue of food safety. Before we can begin to consider potential targets and ways to reduce Fusarium graminearum and Fusarium head blight, we have to look more closely at the disease cycle and the epidemiology of it. The Fusarium inoculum originates from the previous crop's residues. It propagates within there, overwinters, and then propagates under favorable conditions. And in the spring, the spores then spread to the top of the wheat head through wind and rain splash. The wheat head is most susceptible to Fusarium head blight at the time of flowering. This may be due to an increase in reactive oxygen species, which coincides with the most optimal time of pollination for the plant. Unfortunately, this reactive oxygen species also aggravates the fungus, leading to an increased production of the toxin, which functions then as a virulence factor, and it spreads throughout the wheat head. If that grain is harvested, it becomes an issue of food safety. Then the remaining of the plant cycles back in to becoming the next year's inoculum. So within that context of that disease cycle, if we look at the benefits that biochar promotes the health, growth, and productivity of the plant, and enhances the physical properties of the soil. And most importantly, these last three can alter the soil microbial population, alleviate environmental stress factors, and boost disease resistance. We bring that into that disease cycle. We can see that biochar could potentially target, the benefits of biochar could potentially target aspects of the disease cycle and reduce the spread of Fusarium head blight, particularly microbial populations. That's where the inoculum is mainly coming from. Alleviate environmental stress, which both reduces the resistance of the plant, but also the environmental conditions are what increases the inoculum as well, and boost the disease resistance within the wheat head so that it can then defend itself against that initial invasion of the fungus. So in thinking about it, came up with this basic hypothesis that we could collect that crop debris residue, turn it into biochar with a byproduct, hopefully, of bioenergy, and then fortify that biochar with beneficial micro and biocontrol microorganisms and spread it back into the field. The ultimate goal would be to reduce the Fusarium inoculum, but also introduce into that microbial community additional beneficial biocontrol organisms which could compete with Fusarium as the inoculum. While climate is not really controllable, what biochar could potentially give within this system is its porous structure to provide a higher soil retention capacity within the field, which could potentially reduce rainwater pooling and splashing and spread of the inoculum, but most importantly, it would provide a more homeostatic environment versus constant up and down change of water availability. Keeping with that sense of boosted disease resistance, a healthy microbial community also leads to a healthy plant immune system and is also capable of priming the basal defenses so that the plant can more quickly and more strongly respond to a stimulus leading to reduced disease invasion. And finally, though there are few studies on this actually, it is a potential that biochar could increase the total antioxidant capacity of the plant as well, which has been shown in several studies using grape plants in vineyards. This same type of increase in antioxidant capacity can be mimicked by treating the seed with an antioxidant such as ascorbic acid and increase the total antioxidant capacity. The goal would ultimately be to reduce or dampen the production of DON and then the spread throughout the plant. So with these ideas and hypotheses in mind, we developed an initial experiment where we treated a susceptible variety of wheat called Norm with either a control water solution or a ascorbic acid solution following previous methods which had been published, and then planted that seed into two soil mixes, one supplemented with biochar and one without biochar, allowed the plants to grow, and then inoculated them with Fusarium graminearum, and looked at disease progression over time, yield weight per head, and yield weight lost with disease. First, we just looked at the yield of the wheat heads under each of the different varieties, each of the different treatments. And although it has, in several publications, been shown that biochar can increase wheat yield, at least under our conditions, we did not see this increase. But what we did see is a reduction in disease progression and yield loss with disease in the plants that were within soil containing biochar and also had the supplement of ascorbic acid or increased antioxidants. So it could potentially have a synergistic effect between the two. Encouraged by these results, we went on and did a second experiment following very similar type of things with the antioxidant inclusion on the seed, a soil mix with biochar and without biochar. I wish I had heard the keynote speakers talk before I designed this experiment, because I would not have changed the soil to the more ready mix here in this experiment. Now in hindsight, you'll see with the results what changed. But the most important inclusion that I did within this experiment was to include the biocontrol agent of Trichoderma harzianum. And it's another fungus which is well known to compete with Fusarium graminearum. Same thing, allowed the plants to grow and inoculated them. But initially what we did was just wanted to make sure that the biocontrol agent could grow perfectly fine on the biochar treated soil, but on the biochar itself as well. So sprinkled the spore trichoderma dried powder onto the biochar. It did not appear to have any issues in growing. It sporulated, it was perfectly fine. And the plants that we inoculated within the soil had a lot more trichoderma on them all the way up from the root. It was inoculated in the soil. So trichoderma was present in the root as well as in the stem and all the way up to the head inside the florets. We did have some cross-contamination as well in our controls. The trichoderma did spread, but it was evident that the ones that we inoculated had a lot more of the biocontrol agent on it. Looking first just at the yield with the treatments, again, we did not see any effect of biochar amendment. However, we did see a significant effect of the trichoderma. In almost all the soils that contained application with trichoderma had an increase in yield production for the wheat. However, within this different soil, we did not see any significant difference in disease progression, which we attributed to potentially that difference in the soil which we used. So Sunshine Mix Ready Earth, while it has an additional supplement of nutrients and a higher concentration of peat, it gives you more, when you grow wheat in it, it gives you more heads to work with, so I could increase my N number. But even though I did that to try and increase my N number to help with the statistics, I think it just eliminated the effect that was taking place. And from what I've learned here and coming to this conference, that it can make such a big difference, the type of soil you use, and it's not uncommon to see variability from one experiment to the other. So, of course, my work is still very much in the preliminary. I'm in the process of doing toxin analysis and have a lot more ideas of where I'd like to take this. But just for in summary of what we know so far is the combination of ascorbic acid seed treatment and soil biochar amendment enhances wheat resistance to Fusarium head blight, leading to reduced disease spread and grain loss with disease. However, there appears to be an effect of the type of soil that you use, so it probably won't work in every soil in the field. And finally, that biochar can be used as a carrier for the introduction of beneficial microorganisms such as Trichoderma harzianum, which significantly increases the yield. And with that, I'd like to acknowledge all my collaborators, especially Steve Vaughan, who introduced me to biochar and its benefits, my research leader, Todd Ward, and my technical support, Jennifer and Stephanie, which are fantastic. And with that.